Thanks, Morgan. I want to thank everyone for the really warm welcome to the OICR community. As you mentioned, I very recently joined Mount Sinai Hospital in the Lindenfeld-Tenenbaum Research Institute, where our new group is looking to use multiplexed imaging for both the study of cancer progression in mouse models and for precision medicine approach. But today I'm going to be focused more on my postdoctoral work in Baron Bodenmiller's group at the University of Zurich, where we used imaging mass cytometry to study breast cancer. Now, like most diseases, breast cancer is arising from within normal tissues, and it's distinguished from these normal tissues by changes in the organization of the cells and also by the identification of biomarkers. But we know that no individual marker is really good enough or detailed enough to provide information or proper classification of all these tumors. So with more markers, we're actually able to get closer to providing better biomarkers and more information about these samples. With multiple markers, we're then able to look at many cell types at the same time and also the functional states of those cells. What are they doing? What their signaling pathway networks are? Are they growing? Are they dying? And we're not just looking at individual cells because we have many markers, we're looking at multiple cells working together all in the same sample. So my focus has been mainly on breast cancer, which is any tumor arising from within the breast tissue. And spatial information is very important for the classification of these tumors, as the early growth is usually in the alveoli, the milk-producing cells, or occasionally in the ducts that transport milk to the nipple. And the early proliferation stages will fill in the hollow areas of the duct or the alveoli, but it's not actually considered breast cancer until these cells have broken outside of the normal tissue. So spatial information is needed to identify malignant from non-malignant samples. Now, any tumor arising within the breast is breast cancer, but it's known that there's many different types of breast cancer that can arise and differences between the tumors in different patients, which we refer to as intertumor heterogeneity. But beyond that, there's also heterogeneity within an individual tumor, and that different regions of a tumor can be of different types or have different involvements in the microenvironments or differences in immune response. And this is really linked to outcome for these different patients. So these different types of breast cancers can be classified using immunohistochemistry and pathology imaging to classify 4 different types of breast cancer, either hormone receptor positive, HER2 amplified, or the absence of these markers, which are triple negative. And this will help identify which types of therapies these patients should receive. If they express hormone receptors, should they receive hormone therapies or endocrine therapies, or if they have amplified HER2, should they receive a targeting drug such as Herceptin. Now, the worst outcome is actually seen in triple negative cancers, which are just the absence of hormone receptors for HER2, and there's no specific targeted drug available. But beyond this kind of simple classification of different types of breast cancer, we're not really looking at what's going on in these tumors in terms of intertumor heterogeneity. As a tumor that could be classified as having both hormone receptors and HER2, we don't know, is this one cell type expressing both markers, or these 2 different cell types working together to form the tumor, and we need to target both of these with a treatment. Beyond this, when we're looking even at one marker, we don't look at different levels of expression of that marker. We just say, is it positive or negative? And because the most dangerous cell type or the most dangerous tumor type is the absence of markers, if these cells are present in any other tumors, we would never classify this. So we really know that beyond the simple classification we use, there's a lot more complexity happening in these tumors, that there's more different types of tumors. The organization of these is important, and also the involvement of the tumor microenvironment and the immune system. So to really get at a thorough understanding of what's happening in these samples, we need a technology that can have single cell resolution. It needs to be multi-parametric. You got to look at many things at the same time. We need spatial information for the organization of the tissues. And we need to be able to quantify all these markers. And very importantly, it needs to be compatible with clinical samples coming out of the clinic. So for this purpose, the Bodenmiller lab at the University of Zurich had created imaging mass cytometry. So this combines spatial resolution provided by laser ablation with the multiplex measurement capabilities of a CyTOF ICP mass spec. So CyTOF was originally created as kind of a competing technology with flow cytometry, and now we're using it as a competition with fluorescence-based imaging. And CyTOF is able to look at many more markers at the same time, as instead of measuring fluorophores on antibody tags, each antibody is tagged with a different metal isotope of a specific mass. And then these are then read out on a mass spec, and with the very sharp, distinct peaks for the different masses of the different tags, we can look at many, many markers at the same time. We don't have to deal with the same issues of overlapping wavelengths that are present with different fluorophores. So for imaging mass cytometry, we use a very similar staining protocol to immunohistochemistry or immunofluorescence. The difference is that instead of fluorophores or an HRP, we have each antibody tagged with a different metal of a specific isotope. The stained tissues are then introduced into the laser ablation system of the IMC, and the laser then rasterizes the tissue. So pulse-wise, it's destroying a 1 micron spot of tissue or aerosolizing it. This aerosol is then introduced into the plasma of the mass spec, where it's completely atomized. And on the time of flight mass spec, we're measuring all the metal tags that were present in the initial stain. So for each laser shot, we get a 1 micron squared measurement for all these metal tags, and this then becomes a pixel in an image that we digitally reconstruct. So for example, in the recent papers, we are looking at 35 to 40 antibodies all stained on one piece of tissue at the same time, and this allows us to look at markers of different cell types from breast tissue, including the clinically used immunohistochemistry markers such as hormone receptors or HER2. At the same time, we're able to look at markers of different cell types in the microenvironment, such as for blood vessels, different immune cell types. We're also looking at different factors that are known to be altered during cancer progression or lost, such as p53. And we can identify signaling features of these cells, or if they're proliferating, or if they're undergoing cell death. So to give you an idea of what these images look like, this is a breast cancer tumor where the tumor is on the left of your screen, and on the right, you're looking at the healthy fatty tissue of the breast. And what I'm going to show you is a combination of 3 colors, but every single marker is present in the same piece of tissue. The issue is more that you and I can't look at 35 to 40 colors. So we look at them similar to IF, but all the stains are present in the same tissue. So this counterstain reagent is ruthenium tetroxide. It labels all the proteins in the samples, but we're then also able to use markers to look at matrix or proliferating cells, just here with phospho-H3, those are mitotic cells. We can look at the myoepithelial cell layer with smooth muscle actin, or epithelial cells with epidherin, and this allows us to identify where the tumor cells have actually broken through this myoepithelial layer and become invasive. We can also look at methylation marks in the cells, and we can see different types of immune cells and where these are localized differently, where different immune cells are interacting with different parts of the tumor. And at the same time, look at the heterogeneity of that tumor using many, many markers. So to analyze these images, what we do is we define the region of the image that corresponds to every individual cell. So we segment the image, and then we quantify all 35 to 40 antibodies, the average in each of those regions. And we also look at the spatial features of that cell. So we look at its size or its area, how much of its membrane is touching a neighbor, and even the identity of those neighboring cells. And we use all this information similar to how you would use single cell data from suspension. And we use dimension reduction tools such as a Tisney plot that you see on the left. And so in this study, we were looking at over 300 sections of breast tissue on a tissue microarray, and we could identify upwards of 60 different epithelial cell types, as well as different cellular types in the microenvironment. And those are visualized here on the Tisney plot. So we don't just know the identities of the cells, though, we know their location. So one of the things that we can do is look at these cellular phenotypes and their distance to the tumor stroma boundary. So these plots on the right, you have your stromal and immune cell phenotypes on the top and tumor cell types on the bottom. And what you see is for that cell type, you see the distribution of that cell from the tumor stroma front. So as expected, we can see most of the immune and stromal cell types are outside the tumor, but then we can also quantify or identify the immune cell types that have infiltrated into the tumor mass. On the other side, we see most of the tumor cell phenotypes were present inside the tumor mass, but we can also quantify rare single cell phenotypes of tumor cells that have invaded outside into the stroma. So when we're doing this, we are identifying all the cell types in every image from every tumor that we're looking at. So in this stacked bar plot, we've quantified the cell phenotypes in all 300 tumors that we're looking at. The colors correspond to the different phenotypes that we see. So in every case throughout most of the presentation, green cells are immune cells, blue are different types of stromal cells, and then the combination of other colors are different tumor cellular phenotypes. So we can count these in each sample, but we can also label them back on the original image to look at their organization. And what we could see is that most tumors were dominated by a specific tumor cell phenotype. And while some small regions were mixed of different tumor phenotypes, and these were present in all sorts of different organizations, all sorts of different interactions with the microenvironments around these. So to look not just at what cells are there, but how they're organized, we look at all the interactions present in these images by identifying neighboring interactions between cells. And then we then want to identify statistically significant interactions. So previously we'd had to make up our own new statistical test to identify this. So what we do is we look at all the cell types we've identified in the tumor, where they're located, and we look across all of our images and we look at correlation. Are these present in the same images? And this will we show with the square color. But then we want to look spatially which cells are actually touching each other, interacting. And so what we do is we compare which cells are interacting in the real image versus which cells are interacting in a randomized version of that image. So 1,000 times we shuffle the location of all the cells while keeping the structure intact. And then we count the number of interactions in the original image and we compare that to the randomized version. So if there's more interactions between two cells in the real image versus random, by a simple t-test, we'd say this is significantly an interacting, two cell types that interact. And we look at this with the circle color. So we could also say if there's lots of interactions between both the random and the original, such as the green cells here, this would not come up as significant. Whereas if we look, let's say, those red cells, previously we could say these are interacting cells, but we can also say if they avoid each other. So if they never interact in the real image, but they do tend to in the randomized version, we'd say this is avoidance. We see this in blue. And we do this for every single possible interaction between different cell types in these images, and you get one of these large matrices. And this allows us to then identify significant interactions. So for example, we could see that T cells were correlated and were interacting with endothelial cells, so that the immune cells were near the blood vessels. We could also identify that proliferating cells were interacting with other tumor cell phenotypes by the circle color, but there wasn't a distinct correlation there, just by the square color. And we can also look at directional relationships. So for example, fibroblasts in endothelial cells, or maybe parasites in endothelial cells, we could see that the fibroblasts were in the environment of the endothelial cells, but not the other way around. And we could see this in the images where there were fibroblasts supported larger blood vessels in many cases. And this could also be seen for fibroblasts surrounding tumor cells. And we could also use this to identify mixtures of tumor cells that were interacting with each other. So will this pairwise identified different cell types that were interacting with each other, or maybe groups of pairs interactions such as T cells, B cells, and macrophages. We also wanted to look more at groups of cells or communities of cells. And so for this, we looked at the overall interaction network in an image, and we used Louvain community detection, which in the past has been used to identify groups of friends on Facebooks or clusters in cellular network information, whereas here we're using it to identify dense groups of interacting cells. And we did this in two ways, just at the tumor cell level, which tumor cells interact with each other, and also looking at including the microenvironments. So all the cells interacting, but in this case, agnostic to the type of tumor, so more of an immune or stromal cell community. And of interest, we could see that the homogeneous tumors, like those on the left, seem to be made up of an individual type of community, whereas heterogeneous tumors were made up of many different types of communities, but the communities themselves were homogeneous. So heterogeneity itself seemed to be separated spatially within these tumors, even though in very defined regions, cells would be more similar to each other. And so if we looked at the content of these individual communities, on the left, you could see that most of the tumor cell communities were made up of one type of tumor cell. And on the right, we can see the diverse different groups of communities. Now, the reason these are kind of funny Tisney plots for the communities is that we could see communities made up of all sorts of different amounts of cells. So even though it's the same cell type, you could go from small to big communities along one of these Tisney arms. So we then wanted to look at the interactions between tumor community and stromal community. And so this enrichment plot of the bubbles looked at different types of tumor along the bottom, different types of environment on the left. And we could see that certain types of tumors elicited distinct immune responses, whereas the more dangerous tumor types, triple negative tumor types, seem to have more fragmented immune responses and a more immune cells present there than the hormone receptor positive tumors. So one of the big benefits of imaging mass cytometry is that we're able to do the multiplexed imaging in formalin fixed paraffin embedded samples. So these are biobanked tissue samples that have been stored for over 20 years. So we had 20 years of follow-up on these samples that we're investigating the single cell communities in. And so if you looked at the clinical classifications based on immunohistochemistry, there's the 4 different subtypes, including hormone receptor positives here in kind of gold and yellow. These had good outcome, and the more dangerous triple negative subtypes here in green, which had worse outcome than those. But when we looked at the single cell phenotypes, we were able to identify 18 to 19 different types of breast cancers, types of tumors. And these tended to subclassify or break apart the currently used clinical immunohistochemistry classifications. So if we looked at hormone receptor positive tumors using the high dimension data and the single cell phenotyping, we were able to further break these apart. So within hormone receptor positives, we were able to identify a highly hormone receptor positive group that seemed to have very good prognosis, whereas other cell groups, such as this one expressing cytokeratin 7 positive, had very poor prognosis. On the other side, we could look at this dangerous subtype, triple negatives, and we could do the same thing. Most of the groups we identified also had poor prognosis, but we were able to identify a certain type of triple negative breast cancer, which had large levels of cleaved caspase, or had expression of cleaved caspase 3, or was undergoing apoptosis that seemed to have good outcome from this very dangerous clinical subgroup. Interestingly, we could also look at the different microenvironments we identified and looked at their relationship to outcome, and we could identify tumor microenvironments that were related to slightly worse prognosis, such as these groups 4 and 6, which both contained active vimentin positive stroma that were also positive for phospho S6. And this is independent of tumor type, a distinct microenvironment related to outcome. But we also want to go into this spatial information. So we looked at just the relationship between these individual communities that we saw in the tumors and their hazard ratio related to outcome. We could identify many small in size communities that were related to bad prognosis, whereas larger communities that were made up of same cell type but in bigger masses would be related to good prognosis. And this was true in most cases, but then we could see distinct cell subtypes, distinct types of tumors where this was actually flipped. And the large communities were related to bad prognosis and the small ones good. So that was of interest. Further, we could look at multicellular immune interactions and identify some of those that were related to good prognosis, such as masses of macrophages or masses of T cells. We also could identify endothelial T cell enriched environments that were related to bad prognosis, so here in yellow. So while this is looked at kind of individual regions from many tumors, over 300 different breast cancers, we also were interested to look at what happens in the spatial information across very distant regions within an individual tumor. So for this purpose, we went to a second cohort where we had 5 or 6 different very distant regions from one patient, and we wanted to look at the heterogeneity across space. And so within this, we were able to identify the same types of tumors, the same types of single cells. And what we did is we studied the Kulik-Leibler divergence, which what it does is it takes all the images that we've taken from one patient and it creates a synthetic average image. And then it compares each image to this average. And that will give a score for the heterogeneity or the distance from an average tumor to each of these regions. So across all the patients, we could then score the spatial heterogeneity for each region that we looked at. And many tumors had very low regional or spatial heterogeneity, where there was a subgroup of less than a quarter of them, where there were individual regions with large amounts of spatial heterogeneity. And we could identify the types of cells in those regions that were more different than the main tumor mass. And we could also look at which types of tumors seem to be more heterogeneous. What was interesting is that these triple negative, very dangerous subtypes seem to be more homogeneous, and also the hormone receptor high, good prognosis tumors seem to be more homogeneous, where what seemed to have a lot of heterogeneity was kind of this hormone receptor positive, cytokeratin low, dangerous subtype. So potentially in specific subtypes of cancer, heterogeneity could be more related to outcomes there. And this information could be very useful for any cancers that are being diagnosed by biopsy. It's important to know if I biopsy one region, is that type of tumor likely to have more heterogeneity, and we needed to do more biopsies in this case to get an accurate picture of what that tumor is. Or can I trust my single biopsy? So this work has used multiplexed imaging to look at 35 markers in FFPE tissue sections, where we identify the single cell phenotypes, their locations, their interactions, with the goal being to basically quantify tumor structures based on many cells being present in there. So it's a multicellular definition of these tumors. And we've shown that this information can really provide additional information on top of what the clinics are currently doing to help identify high-risk patients. So the next step is we want to know how is this related to genomic subgroups. So we can see that we provide more information, but how is this linked back to different types of mutations? So for this purpose, we collaborated with Carlos Calvas's group at the University of Cambridge, where we did this analysis in the METABRIC cohort of patients. So this is a breast cancer cohort, which iteratively over the years, people have added new information to. So they have from this cohort, there is full transcriptomes, there's sequencing of about 160 different oncogenes, there's microRNA profiling, copy number aberration studies. Now what we're doing is taking the fixed samples and using IMC to identify the single cell populations present in these tumors. So within this, we're able to identify very similar tumor and immune cell phenotypes based on the expression of known markers and additional markers in these antibody panels. But now we can relate this back to the transcriptional information, the genome information. So on the left, what we showed is we looked at different cell types and their relationships to different changes, different gene changes, and we could see that there were major differences. Different cell types were related to more different genomic changes. And when we looked at what transcriptional changes were taking place, we could see kind of certain go terms coming out a lot. Cell terms related to cell cycle, or those related to extracellular matrix or immunity. And this was highly correlated with the cell types that we identified in the images. So more T and B cells present in tumors that had transcriptional changes in immune-related genes, myofibroblast or fibroblast populations being identified in tumors where there were changes in the ECM, and then proliferating cells, Ki-67 positive cells being related to different go terms for proliferation. So if we look at the different types of tumors that are defined by the genome, mostly by copy number aberration, there's 10 different types of breast cancers here. And within the genome-defined breast cancers, what we can see is a high correlation or relationship to certain single cell phenotypes. So these are 10 or 11 different genome-defined breast cancers. So intercluster 10 down here on the right is highly related to triple negative breast cancer, this dangerous breast cancer. And this one, it was interesting to see there's still a lot of cellular heterogeneity in there. as we're identifying many different cell populations. Whereas in some of the hormone receptor positive genome-defined groups, we could identify distinct epithelial populations that were present. Further looking at the microenvironments, we could see that myofibroblast populations seem to be present in the more indolent or low-risk hormone receptor positive tumors, and whereas some fibroblast populations were related to higher risk tumors, and that there were more immune cell types present in these as well, so that there was a difference in microenvironments in different low and risk genome-defined tumors. So going further, what we did is we looked at correlations between different genome aberrations or different mutations and these specific cell types. So what you're looking at here is that each row is a specific single cell phenotype we identified, and each column is then a specific mutation or a specific amplification or deletion. And we're looking at correlation between the presence of the cell type and between these different genome aberrations. And we identified things that were well known in breast cancer, such as hormone receptor negative cell types were related to p53 mutations, so you can see down here on the left, whereas hormone receptor positive were more likely to be related to a PI3 kinase mutation. So here, red indicates a positive association and green is a negative association. And the dark black circles are those that were statistically significant. So beyond just the known PI3 kinase hormone receptor positive, p53 hormone receptor negative, we then also identified some very interesting things. One example would be that hypoxic cells seemed highly related to genome aberrations related to immune therapy. So we saw deletion of beta-2 microglobulin related to hypoxic cells, and we saw amplification of PD-L1, which so very interesting new things to kind of go after. So overall, we're kind of looking at tumors as single cell features, and then this more recent work is kind of looking at how the genomic structure relates to the multicellular phenotypes that are present in the tumor and the associations between specific aberrations and specific tumor phenotypes and how this is related to the kind of these patients. So I really need to thank Jana Fischer, who I worked with on the Nature paper and who was responsible for identifying algorithms to look at communities, and Raza Ali, who had come from Carlos Calvis's group and joined us in the Bodemiller lab, who was analyzing the genome to single cell features. A lot of work was done with the pathologists at university hospitals in Switzerland. I also need to thank the patients that donated these samples over 20 years ago. It's really interesting to see you don't know what's going to come from these, and very useful, very helpful for them to have done that so long ago. So we're now a new group at the Lindenfeld and part of OICR, where we're studying multicellular features and looking at their involvement in model systems as well as tumors. And we're hoping to link up with many people in the OICR community to study these things going forward. So I hope to answer any of your questions. Thanks.